Agnieszka works as a senior scientific advisor for risk assessment of genetically modified organisms at the National Food Institute in Denmark. She is responsible for providing science-based advice to government agencies, namely the Danish Agricultural Agency, Environmental Protection Agency, and Veterinary and Food Administration. Additionally, she participates in scientific committees and working groups of EU bodies when called upon by the Danish agencies. Researching and lecturing about risk assessment of GMOs is an integral part of her job. Agnieszka is a molecular biologist by training and holds a PhD degree in genetics and bioinformatics. She calls herself a gene technology enthusiast, and her favorite molecule is DNA. Thanks to her passion for communication and public speaking, she had the privilege to deliver a TEDx talk in March 2022 about safeguarding the most precious DNA source we all possess, our DNA. She's a busy wife and a mom of 2 girls who enjoys cooking and anything creative. To get the most of her me time, she dances and she takes care of her kitchen garden. Butterflies and moths are her favorite creatures. Ladies and gentlemen, Dr. Agnieszka Podolska-Charlery.
Thank you so much for that kind introduction. And thank you all, my dear audience, for being here today, and online audience as well, to participate in this lecture, this interaction that we will have today, where I'm hoping your curiosity about GMOs and your thoughts about GMOs would get ignited, and we can have a really good discussion about this topic after I present some aspects of science around GMO. I used to be a researcher, an active researcher, researching basic science in the lab, and the opportunity knocked on my door, and I decided to switch to a more advisory work, where I use all my knowledge into a much more applicable way, where I advise Danish authorities on GMO-related issues, or sometimes representing Denmark in scientific communities, even though I'm not Danish. So that's quite fun. Today, GMO are such a hot topic. So many opinions, so many feelings about the subject, so many political decisions around it. But from my side today, I will present what science says about GMOs and their impact on our life. And this is a vast topic. And today we only have about 45 minutes for me to talk about it, so we're only going to touch the surface of it and hopefully discuss some more in the discussion session. And I also want to mention that since it's such a huge area of science, I am a growing expert in parts of it, but not all. I'm still learning, and not every aspect of genetically modified organisms world is my forte. So I'll try to answer questions as best as possible, but I also have my limitations in this area. And as a short disclaimer, I am here as a passionate scientist and enthusiast of this technology, and a professional in the GMO field. However, what I say today will be with a pinch of my own opinion and take on GMOs, so this does not represent the view of the EU or the Danish authorities, even though it almost always aligns with what my colleagues think and the Danish authorities, but it's also with a bit of my own opinion on it, professional opinion. Yes, so I'll go through some basic definitions that are related to this subject, because when I listen to presentations and I get lost because concepts are not introduced, I think it's quite frustrating, so I would like us all to be on the same page, and I'll introduce a few basic definitions in this quite technical world. Then we'll tell about which organisms can be modified and how. And I'll move on into the impact on our world as humans, and touching a bit upon science and regulation, with focus on environment and safety of GMOs. Then I'll go into, I find, the most interesting part of the talk, where I will showcase a few examples that, in my opinion, are most impactful. And at the end, I'll summarize what I spoke about today, and I'm hoping for a very heated discussion after that, where we can discuss pros and cons and what your experience is and what you my dear audience, think about that. So, at the center of genetically modified organisms world is DNA. This is our genetic material. It's featured in every living organism, and it can be modified and it can change because it simply has this ability to make mistakes and still be a functional molecule. This researcher that wrote a book about lives of a cell, he thinks that if it didn't have this ability, we would all be still swimming as simple bacteria in the ocean. But because it can change and get better and create a new form of life, then that's why researchers and humans also learn to modify it to our advantage. By far, the most marvelous and my favorite molecule by far. I work for a country that is an EU member state. That's why some definitions I will introduce are by the European Union. However, they do not differ much in other countries. So that's an updated map with UK not being in green after Brexit. Organism is defined as any biological entity that is capable of transferring genetic material. So once you can multiply and give your genetic material to the next generation, you are an organism. Now, genetically modified organism is any organism with the exception of human beings. They cannot be called GMO, according to the EU. It's an organism in which the genetic material was changed, but not in a way that could occur in nature or through mating or recombination. Recombination meaning exchanging genetic material. Okay? So there has to be an interference from a human in order to make a GMO. Some more definitions. So genetic engineering, which also is called genetic modification, is a process where a variety of laboratory-based techniques are used to actually do that, to alter a DNA of an organism, to introduce or change something that will end up being advantageous. And in the EU, all these techniques are divided into established genomic techniques and new genomic techniques. Genomic techniques meaning techniques working on our genome, our genes. And that all has to do with EU adopting a GMO deliberate release regulation in 2001. So established techniques are all those that existed and were widely used before 2001, and new genomic techniques, all those that were established and are now established and used after, or came up after. So I want to say that just shows you how long genetic modifications of organisms has been happening. It is not a new technology, a new way of producing organisms at all. But now, let's get to it, the technology. I'll give examples of different organisms, how it is done, and please, if I say something that does not make sense or I did not explain properly to you, please interrupt me and tell me to explain in another way or again. I'll be more than happy to do that as I speak. So now let's start from this one of the simplest organisms, a bacteria. Bacteria have, I don't have a pointer. Bacteria has two sort of sets of genetic information. It has a main chromosome, that is this curly thing that says B. A shows a bacterium, then B is its main chromosome, and a C is a circular piece of DNA that every bacteria has, and it is used to exchange information and give useful genes from one bacterium to another. So they actually can trade it. Whenever they find something is useful and make them survive, they want to give it to their mates, so they would share it. But scientists also found a way to take it out of the bacterium, modify it, open it, and put different things in it that have never been in the bacteria before. So now, once it is modified, a new piece of DNA, a gene that perhaps we are very interested in, is put in that circular form. It can be pushed back in the bacterium, they will multiply and produce that that protein or that enzyme or whatever it is that we really wanted them to. And bacteria grow to very high numbers in a very short amount of time. And it's rather cheap to grow them on commercial scale in fermenters and so on. And that technology was used to produce one of the most precious substances humanity is using nowadays, insulin. Insulin is a medication given to people with diabetes. And this is exactly how it is produced. It used to be produced in E. coli and still is. E. coli is a simple bacteria that lives in our gut and in guts of other organisms. It can cause disease, but it can also be modified to produce a human gene. So imagine a bacterium now gets a human gene put in it, and it will produce a lot of it, and we can purify it and use it to treat humans. So here we see a human insulin gene, this circular form of DNA from a bacterium that is opened by researchers. Then the human gene in orange is put in the circular DNA form, and that all is introduced back in the bacterium that is grown in huge fermenters to huge quantities and producing insulin that is then purified and put in a bottle for the patients to use. And why is it so important? It is important to be able to produce huge quantities of insulin and rather cheap, because only in Western Pacific region, which is China, Australia, and some other countries in that region, 206 million people suffer from diabetes type 1 and 2 combined. North America and the Caribbean region, 51 million people suffer from diabetes. And insulin is very much needed. A GMO is producing insulin, but insulin itself is not a genetically modified organism because it is just a protein. It's not an organism. Now, for agriculture, modifying plants, genetically modifying plants is very important and widely used. And again, science figured that a bacterium called agrobacterium that causes sort of a cancer in plants can be modified and used to introduce, instead of causing a disease, it can actually introduce a gene that we are interested in into a plant, that being resistant to a bug, a pest, or other traits such as higher yield and so on. And this is achieved because, again, there is a circular TI plasmid, circular piece of DNA that has a tDNA that upon infection of a plant cell gets introduced into the plant genome, integrated in it. Bacteria are so clever. They can do so much. And using various techniques in the lab, such infected cell with now integrated piece of DNA that we are interested in, the modified cell can be now grown in a culture in the lab using different factors that will create a plant from one cell. So these are cultured plant cells that will then give rise to a modified plant that can be then reproduced. And actually tobacco is it is quite easy to modify tobacco in that way, but other plants too. So now, when it comes to such modifications and comparing traditional breeding of plants with plant biotechnology, meaning plant modifications, you can see in traditional plant breeding, we have a desired gene in orange in a donor plant. There is a plant somewhere that has a very high yield, for example, or we find it has a feature that we are very interested in. And then a plant breeder, has a commercial plant variety that they would like to improve. What they would traditionally do, they would cross these two plants, if they can be crossed, right? They have to be related. They would be crossed, and then a shuffling of genes happens, pollination happens, shuffling of many genes happen, and then some of them will have that gene of interest. And then the breeder will select, and select until they have a new plant. With plant biotechnology, We still have a gene that we are interested in. We still have a commercial plant variety. But to move that gene to create a new variety, improved one, it is a lot more precise because we do not do any random shuffling of genes. We take what we want and we put it, only that, directly in the plant. Traditionally bred plants, no risk assessment, only field trials to see if they yield well or if they are not very susceptible to drought or pests. No risk assessment, easily approved. GMO plants, at least in the EU, heavy risk assessment, very, very strict rules, and no approval. GMO plants are not grown in the EU. Other parts of the world approach it differently, but in the EU, plants come only as import and mostly, almost exclusively, for feed for animals. Countries like China, US, India, some countries in South America, South Africa, and for example, Burkina Faso, a very different approach, but that's the EU. And now, last organism that is my specialty. I mostly work with genetically modified viruses. All that is done to deliver medication to humans and to produce GMO-based human medication. So researchers take a scientist, a pharma industry, takes a virus that is harmless to humans. They manage to put a gene of interest, human gene that would help with a disease, genetic disease that we might have or humans might have. They will package that good gene in a virus, deliver it to humans to hopefully cure the disease. So it's all about, there are many, many genetic disorders in humans. Some very severe, some less severe, and even many cases of blindness are caused by various genetic disorders. So it is very important that medication to help people that are touched by these diseases exist. So healthy gene in a viral vector that will be delivered to humans with minor difficulties and hopefully help curing the disease. And there are two ways of looking at it. We call it in vivo. That means where a good gene is packaged in a viral vector that actually does not have many of its own genes, and it's delivered directly into our bloodstream, so it will be distributed many places, or actually it can be delivered directly into the brain or to the eye and so on, so it can be very targeted. Ex vivo, meaning outside our organism, is when normally with blood disorders, cells can be harvested, modified in the lab, multiplied to big numbers, and put back in humans to help with different blood disorders or blood cancers and so on. Two different ways. Again, this is GMO doing the job. And actually, another example is GMO-based vaccines. There are many vaccines used in poultry industry that are GMO-based and approved widely all over the world. And the recent example is 2 GMO vaccines for COVID that we had approved. And that actually, I think it's most interesting example because that's the first case I ever risk assessed. It's a Ebola vaccine. Ebola is a terrible virus infecting humans. Even now, with some medicine and very good care, 50% of cases are dying after contracting the virus. It can be given to humans by bats or non-human primates, and then it spreads like fire, even from dead people, because it's present in saliva, in semen, in blood, in feces, in all the bodily fluids. So once a person contracts it, then it's an infection of many people very fast. And a company figured how to create a vaccine against it. And funny enough, VSV virus is a vesicular stomatitis virus. This is not a human virus. It's a virus that infects pigs, cattle, and so on. It is harmless to humans. And then they took one gene from a Zaire strain of Ebola virus, put it in the vesicular stomatitis virus, grow it to high numbers, and that was given as a vaccine to humans. Interestingly, farmers who receive this and work with animals on day-to-day basis, they have to stay away from the animals for a few weeks post-vaccination in order not to risk infecting the animals, because it is a vesicular stomatitis virus with some genes removed and some genes added, but the virus still has capability in theory, so there is a risk. In risk assessment, word no risk does not exist. If anything, we can say that the risk is negligible. But in this case, this was one of the risk minimization measures for that product. And it was also used as a compassionate use protocol, meaning that Doctors Without Borders and people sick from Ebola in Africa were given this vaccine even before their market authorization was issued, because when the disease is so severe, it is allowed. Now let's go to the impact, and I'll focus here a little bit about the science and the regulation. So in the EU, the political decision has been made a long time ago that plants will not be, GMO plants are not grown in the EU member states. They can be imported for feed mostly. And plants that are imported to the EU are mostly soybean, maize, rapeseed oil, and cotton. And the traits that are of most focus and are featured in those plants is insect resistance, herbicide tolerance, and the changed fatty acid profile. So rapeseed oil exists that has a little bit more beneficial fatty acid profile, and then pollination control is one of the GMO plants' focus. Now, the risk assessment is all about assessing the risk and managing it. Managing the risk is political. Assessing the risk is what scientists do. And when it comes to plants in the EU, European Food Safety Authority deals with that. When it comes to medicine, European Medicine Agency is at the center of it. So my job as a risk assessor is science-based. So I need to know the regulation, but I most definitely need to know what science says about GMOs and all the newest achievements. But very importantly in Denmark, stressed a lot, it is politically independent. So managers, on top of science-based risk assessment, have to apply what society will think, what is a good political decision, but I am not influenced by that. All I do is I deliver the assessment based on scientific evidence. And I'll focus on plants because I think this is somewhat closest to life and most relevant. The risk assessment, at the center of a risk assessment of a GM plant is pretty much a comparison. How much does this plant compare to the non-GM comparator? All of it happens in that kind of way. So then we have conventional plants, and then we have GMO plant. And it is almost always case-by-case basis. They are very different modifications. They are different plants and so on. But what we focus on is that we assess the molecular biology of such new product, such new plant, meaning we look at the DNA. Where did the modification happen? How well it is described? If it happened many places in the genome, if it is disturbing any important genes in the plant and so on. So that's what we look at. Then we compare the yield to a non-GM comparator, or even if the plant is a lot taller or a lot shorter, very simple traits. But the big part of risk assessment of a GM plant is toxicology. So they are studies made in rats. That's how it is in the EU. This is the law that a company must conduct a 90-day study, feeding rats for 90 days with their product, and then assessing, comparing to the control if any changes happened. Allergenicity profile must be assessed because, for example, soya causes a lot of allergies already. So if you introduce yet another protein in it, it might cause even more allergy, and you don't want that. So that has to be assessed, and the nutritional value. Because dietary guidelines are made on conventional plants and what they give us. So if you all of a sudden disrupt the amount of nutrients a plant gives you, you have to maybe adjust the dietary guidelines and so on. And then another step 4 is the assessment of a safety to the environment. So far, there has not been one scientific report coming from any part of the world based on literature and science and evidence that says GMO plants are more dangerous to the environment than conventional plants. Such report doesn't exist. So there are many skeptics of GMO, and that's understandable and so on, but science simply cannot find evidence how these plants in any way can be more harmful to human health or the environment than conventional plants we have right now. A lot is changing now in the GMO world. EU, there's a lot of pressure from various stakeholders to adjust the GMO regulation in the European Union. US is a lot more relaxed. Lots of GM plants are grown there and sold and consumed and so on. Many, China, India as well. EU is very strict on it. But what I think is very interesting is that Kenya, actually in October this year, just lifted a ban on GMOs. So that means now they will allow GMOs to be grown and sold in their territory. And of course, this was welcomed by some, but also opposed by GMO skeptics. And then again, issues to human health and environment were quoted. However, a World Bank policy research paper and many reports made by different bodies point that actually genetically modified plants can benefit agriculture and humanity, and securing that enough food is produced, and they can offer a powerful tool for nutrition. Then I'll talk later about a golden rice case, which is my personal favorite genetically modified plant. And then they can also help farmers to adapt to climate change, because the hopes for genetically modified plants are that they will be drought resistant. Now, before 2012 ban, Kenya was not so strict on GMOs. However, in 2012, a study by French researcher came showing that a study in rats showed that consuming GMOs can lead to cancer. And based on that study, Kenya banned GMOs for good for 10 years. That study then was taken to pieces and criticized by the scientific community, and even from our institute there was a letter written to the editor saying how wrong the design of this study was, and how controls were missing, how statistics was not done properly in the results. And at the end of the day, shortly after, the study has been retracted. It can still be found online, but it is a retracted study. However, Kenya never changed their opinion on GMOs after that. So that just shows you what kind of impact science, done properly or not properly, can have on big decisions that countries make. Now I would like to move to the products. GMOs are everywhere. They really are. Lots of enzymes for laundry are produced by genetically modified yeast or bacteria. Again, US grows tons of genetically modified food. And I'll present you a case of a corn, genetically modified corn. It's called MON 810, Monsanto 810, that developed. The story is actually quite fascinating. So at some point, farmers in the US noticed that the corn cobs were getting infected and covered in fungus. And they were losing a lot of yield from that. Then they noticed that some plants had kind of a grayish leaves, and they were a lot more resistant to that problem. So what they did, they took the gray leaves, they rinsed it in water, and then they used that water to spray their fields that were not gray, and that helped. A lot less insects were biting the corn, and then the fungus was not infecting the corn as much. So researchers picked up on it, and they figured that there was a bacterium that was producing toxin that was killing the bug, the pest that was causing all the problem. And then Monsanto created maize that produces that toxin. So even when the bug wants to start eating it, it will take a bite and it will die, because the plant is naturally producing that toxin. So here you can see how the pest looks like. These are little moths. And then Monsanto 810 maize versus a non-GM comparator. And I mean, this cannot even be used for animal feed, because fungus can produce lots of toxins that is to be discarded. So a massive loss in yield. And another interesting example actually is banana. Two issues in two regions of the world. In Africa, there is a struggle with bacterial wilt, and then actually banana plants, transgenic, modified banana plants were created, where a gene from pepper was given to a banana plant, and now banana was resistant to that bacteria, because that was a gene in pepper. But this bacteria never infects pepper plants because they are resistant to it. So now they created banana plants that are resistant. And in another part of the world, actually in Australia, I believe, they had a different problem where banana plants were dying from a fungus infection. And researchers found a very old wild variety of banana that was resistant to it. They found the gene responsible, put it in the new variety, and that's how they achieved some lines that are now a lot more resistant to that fungus. And golden rice. So golden rice, as you can see, it's not white. It's orange, yellow. And that's because rice normally does not have any vitamin A in it. And in golden rice does. And that's its biggest advantage and beauty. Because in regions like Southeast Asia and also some regions in Africa where sometimes people pretty much only rely on rice in their diet, many children die of vitamin A deficiency or blindness. They get blind because of vitamin A deficiency. With providing a solution that can actually give children vitamin A in sufficient amounts, could save 2.7 million children globally from dying or getting blind. To me, now my personal opinion coming, to me, this is fantastic. This is fantastic. When golden rice trial fields were being conducted in India or in other countries, Greenpeace and other big organizations were destroying them completely because they are against it, and this is fine. But thinking, and this is a technology that I believe a German scientist figured that it's not patented. He didn't want to patent it. He wanted the world to benefit from it. So basically, he activated a vitamin A production pathway in rice. He modified it in a way that now it's producing that. And in 2019, Philippines allowed to grow golden rice. So now in the Philippines, it can be commercially grown, and also in the US and Canada. And that was approved a little earlier. So the amount of vitamin A in golden rice too is somewhere between squash and tomato. And skeptics say, yeah, but it is low. People could eat other things. But the problem is that people who need this, they cannot eat other things. They don't have a varied diet. They don't have a very healthy diet, very varied, healthy, nutritious diet. Therefore, providing vitamin A in the food they do eat is a great solution. Then in the green box, you can see that in 2020, 94% of soybean grown in the US was GM, and 92% of corn. And there are many different GM plants that are actually genetically modified and grown commercially, not in European Union, in many other countries. And I'll go back to some examples of that. But I find in the animal world, we don't have so many examples, but we do have Aqua Bounty salmon that has been 30 years in the making. And I believe in 2020, it was approved by Food and Drug Administration in the US to be sold. This is a salmon that has a genetic modification where a gene responsible for growth hormone is from another fish. And a little element called promoter that is helping that production is from yet another fish. So they put it in a regular salmon, and thanks to that, the salmon grows all year round and not only during spring and summer. Therefore, market size is achieved in about 18 months instead of 3 years. So these fish you see at the top, they are same age, and the GM salmon grows simply faster. And FDA, based on scientific evidence, says that it meets the requirements for safety and effectiveness. That's why it can be sold. And these fish are not grown in any aquacultures. They are grown in massive tanks inland, very far from any water source. And even the eggs and little fish are kept somewhere else than the grown-ups are grown. So this is fish that is most tested, most checked, and yeah, if you want to eat a fish without any heavy metals in it, then you would go for that for sure. Now, sickle cell disease. I find this relevant because, in fact, a sickle cell mutation that is causing sickle cell disease happened a few thousand years ago in Africa, where malaria was also present, because, in fact, a normal red blood cell should be nice, plump, and round with a big surface so it can carry a lot of oxygen for us. Sickle cell, red blood cell, is a very different shape. And in fact, this protects from malaria because the parasite needs a lot of oxygen and a nice big erythrocyte. So if they don't have it, they cannot multiply as good. So, however, this genetic disorder causes people to feel tired and causes a lot of pain because these cells, they clog our blood vessels, and it's a really massive problem. And it is a hereditary disorder, meaning it goes from parents to children when two affected copies meet in an offspring. And about 300 children a year are born with this disease, and worldwide it affects in most people in Africa, in sub-Saharan region, over 5 million people. In Europe, not so much. It's simply because, but it's getting more because people migrate more, and so on. So lots of people are affected by this. Right now, so to the left, you see drug treatment. This is pretty much, there is a drug that can be given to just help with the symptoms, to prevent the cells clogging the vessels so much, to help a bit with that. Then there can also be a transplant, a bone marrow transplant, that from a donor to a sick patient, where they can get cells in the bone marrow that will produce proper red blood cells. But you never know if you're going to find a donor. It's a very expensive procedure and so on. Then another procedure is ex vivo gene therapy. So as I mentioned before, a patient with sickle cell disease, some cells that can become blood cells can be harvested, modified, so they don't have that mutation anymore, and they can be given again to the same person. So rejecting transplant is not an issue because it is your own. However, to prepare your body for that, you kind of have to kill your own cells in your bone marrow with chemotherapy first. And that is very harsh. So no treatment is approved yet. It is all in clinical trials and in preparation because they need to find a better way for that. And then one of the new genomic techniques would be to actually deliver a machinery that will go to your body and modify the bad cells in your body, change them into the proper genetic code, So then none of that preparation is needed because all the cells, or most of the cells that are with genetic mutation would be corrected. But that is in the future. It is not available as yet. And now plants. So this is from a FDA page called Feed Your Mind. There are many examples of genetically modified plants. So this is a glyphosate resistant beet. So glyphosate resistant is basically they created a plant that will be resistant to a herbicide. So you have a field with commercial crop that you want, wheat, beets, and lots of weed growing in it. You have to kill the weed in order to get a proper harvest. And these plants resistant to glyphosate allow farmers to spray glyphosate, doesn't matter which growth phase and so on, and they will kill the weed, but they will not kill their commercial plant. And it's because I believe there is an enzyme in the plant that makes it resistant to that herbicide. This is one of the first commercially grown and widespread GMO plants. It is so unfortunate that this was the first one because it sparked so much negativity towards GM plants, which is understandable because, of course, then some studies shown that glyphosate can cause cancer and it's dangerous to humans and animals and so on. So by introducing a plant that it's created for a company to sell particular product, it was just very unfortunate that was one of the loudest cases, because if golden rice was, I strongly believe the way we look at GMOs would be very different, but it is what it is. Then we have an apple. Its shelf life, it's 28 days. And when you cut an apple or you just knock it lightly, it just gets brown and ugly. So that's not pleasing to a customer, but this one can survive a lot more on the shelf. When I presented that to some students at the course, my colleague, that is a toxicologist, he says, oh, I will never buy that. I will not eat an apple that was sitting in a store for 28 days. I don't want food like that. And fair enough, fair enough. But he lives in Denmark. He has the luxury to maybe even have an apple tree in his garden if he chooses to. But not everywhere in the world people have the luxury to eat fresh produce every day and have access to it. Now we have potato. With potato is a little bit different story, because when you fry or heat treat potatoes, Irish potatoes, they produce acrylamide, which is a very dangerous and carcinogenic substance. And this variety, always. So McDonald's has, even our institute issued guidelines to restaurants how long and at which temperature they should fry their fries to minimize the amount of acrylamide created. And this variety will have less of it when heat treated. So that's the modification. And now, maybe you'll find this example quite relevant. This is papaya, popo. And that's popo. And that's a ring virus causing the disease in papaya. And it almost destroyed papaya industry in Hawaii quite some years back. And I believe it all started in 1990s, where almost all regions of papaya, where papaya was grown in Hawaii, was affected by this. And farmers were losing close to 50% of their harvest, and papaya is the second largest export for Hawaii. So then a government of Hawaii and researchers joined forces to create a virus-resistant variety. And it was a success. In 1998, the government approved growth of genetically modified papaya called rainbow papaya. And I believe 2 years after, 50% of papaya grown in Hawaii was this. And now 90% of papaya is genetically modified papaya, and they can produce and export. So the only issue with this product was that it took them 10 years to get it approved in Japan, which is one of the biggest purchasers. So it is tricky, You have a product that you have to, in a way, use, because otherwise you have to switch to growing something else, or you cannot do where you get your money from. But then someone will not buy it. So this is a very tricky situation also with GMOs. And I'm coming to an end of me speaking. And I just want to say that looking at genetic modifications, it is still DNA. It is only DNA that it's different, and we all have DNA in us, and humans have been modifying plants for generations, for thousands of years. What we eat now and we call cabbage, cauliflower, or broccoli was this many thousands years ago. And this is a weed in Poland. We as children, we would pick, now you see flowers, but when it makes seeds, it makes cute triangle seeds, we would pick it and eat it for fun. But this is where cabbage comes from. And it's not because all of a sudden nature decided, okay, I want cabbage now. I'll cross and make cabbage. Humans did it using variety of techniques. The same with corn. Corn used to be a grass. So it is nothing new that we alter plants to be what we want them to be. And to summarize, I would like to say that a wide range of organisms have been genetically modified for decades now. This technology is not new. And it's developing faster with very new ways of editing genomes and changing genomes in a very targeted way. The approach to GMO regulations differs greatly from country to country and from region to region, EU being very tight on that front. And GM technology, oh yeah, I said that already, is developing fast. And they will become a part of our life sooner or later. I strongly believe so. And seeing at what is happening in the world and in the regulation, it will happen. In the EU, very slowly, but it will happen. And most important from me is that science says there is no reason to fear this technology. This is what science says. So thank you very much for listening.
We have our Vice Principal, Mr. Kid Harris, to present a token to our esteemed speaker.